Welcome back to the Deep Dive. Today, we are on a mission that feels, well, a little like science fiction. We're tearing apart the whole concept of aging.
It really is a fundamental shift.
For so long, we just accepted aging as this, inevitable decline. But the science we're looking at today treats it as a solvable software problem.
And that analogy is just perfect. It changes everything. When we say a software problem, what we mean is the hardware, your actual DNA sequence, is mostly fine.
It's intact.
It's intact. The problem is the instructions telling the cells how to use that DNA. That's the epigenome. And it gets corrupted, like an old computer's operating system just full of glitches.
And what's really astonishing from the research we've dug into is that this software corruption isn't just cosmetic, it's the root cause. It's the thread connecting heart disease, diabetes, cognitive decline, all of it.
Exactly. And if you can target the aging process itself instead of just treating each disease one by one after it shows up.
You could prevent them all at once.
In theory, yes, you could. So today's mission is really a crash course in how to reverse that corruption. We're going to unpack the new tools scientists are using to literally measure your biological age.
Then we'll get into how this information loss happens at a molecular level.
And finally, we'll look at the therapies from gene editing to chemical cocktails that are being developed to hit a cellular reset button.
This deep dive really shows a world where aging itself becomes the main clinical target. And, it feels a lot closer than most of us think.
Okay, so let's start with measurement. If aging is the problem, you need a really precise speedometer to track it.
Right.
For years, we've had these DNA methylation clocks, but they're evolving into something much more powerful.
Which brings us to this idea of a full life cycle clock. One of our sources introduces something called EHR former. Even the name sounds futuristic.
It is. The critical thing about EHR former is that it's trained on electronic health record data, so it's not just one snapshot.
It's everything.
It's everything. From birth all the way to old age, it pulls in data from multiple organ systems, your blood profile, immune markers, how your liver and kidneys are doing.
To give you a much more accurate picture of your biological age, your BA.
A far more accurate picture than just your chronological age, your CA, yeah.
I thought the detail on pediatrics was fascinating. The sources said that for kids, the standard definition of age, accumulating damage is ill-posed.
That's a great point. It doesn't make sense for a child.
How can they be accumulating damage when they're growing so fast?
Exactly. For a child, all that change is program development. It's not breakdown. So in pediatrics, the term changes from an aging clock to a physiological maturity clock.
Which makes so much more sense.
It does, and it lets doctors spot things like maturation precocity or developmental delays or even malnutrition way earlier than before. It's a game changer for early intervention.
But for adults, the big takeaway is the age gap. That difference between your biological age and your chronological age. That's the number that really matters.
That is the ultimate predictive tool. If your biological age is accelerating, that's a critical biomarker for risk. And the accuracy of this new model is, well, it's kind of staggering.
How staggering?
For predicting things like coronary artery disease or diabetes, the model scored an AUC of 0.976.
Wait, 0.976?
Yes.
That's almost perfect. What does that number actually mean for someone listening?
It means we're getting very close to clinical certainty. An AUC score that high means if this clock says your biological age is, say, 10 years older than your chronological age.
Your risk for that disease is through the roof.
It's drastically higher, yes. And it allows a doctor to intervene maybe a decade before you would even show symptoms. It's true preventative screening.
And this leads to another idea, which is that aging isn't, uniform across the body.
Not at all.
My liver could be aging faster than my heart. So a single number misses the details. And that's where we get to systems age.
Systems age is all about that heterogeneity. It's a specialized test that quantifies aging in 11 different physiological systems, all from a single blood test. And what's so powerful is that these system-specific scores are often much better predictors than one global number.
Can you give us an example of that? What really stood out?
The specificity. The heart score from the test was a really strong predictor of myocardial infarction. The lung score was a great predictor of lung cancer.
Even after you account for things like smoking.
Even after adjusting for smoking. That's the revelationary part. You're diagnosing a system-specific vulnerability from a blood marker, maybe before any real damage has set in.
Wow.
And you can see the biology in the data. The heart and lung scores were really closely related, which, makes total sense. They share vulnerabilities.
Sure, things like smoking or circulation.
Exactly. And the liver and blood scores were also tightly correlated, which again makes sense given the liver's role in filtering blood. It just confirms that aging is more like a constellation of local fires, not one big bonfire.
Okay, so if the clocks tell us how fast we're aging, let's get to the why. This brings us to the information theory of aging. I really love this software problem metaphor because it just screams hope.
It does.
You can always reinstall the software.
That's the whole leap. The theory defines aging as corrupted epigenetic information. But the key premise is that this corruption is, and this is the important part, reversible. The epigenome is that instruction manual that tells a cell, okay, you're a skin cell or you're a neuron.
So, how does the manual get corrupted? What's actually causing this epigenetic erosion?
Well, the main culprit isn't what you'd think, like toxins or something. It's the body's own repair processes. Specifically, its response to double-stranded DNA breaks, or DSBs.
And those happen all the time, right?
Thousands of times a day in every cell. And every time the cell has to repair one of these breaks, it has to reorganize the chromatin, the bundled up DNA, right around that break site.
And that repeated reorganization has a side effect.
A disastrous one. You repeat that process millions of times over decades, and you get what they call epigenetic entropy, a loss of organization. Imagine a librarian who keeps fixing torn pages, but every time they put the book back on the wrong shelf.
So all the books are still there?
The DNA is still there, yes. But the library's catalog is a complete mess. You can't find anything. That loss of structure is epigenetic entropy.
And there was this incredible proof in mice with something called the ICE system. They showed it's the repair, not the damage itself, that's the problem.
The ICE system was just brilliant. They designed it to make these cuts in the DNA that were always repaired perfectly without any mutations. The whole point was just to stress the organizational system.
Just to make the librarian run around the library over and over again.
Precisely. And they proved the theory. It's not about what the damage does to the code. It's about how the repair process scrambles the instruction manual.
So what happened to the mice?
The damage dramatically sped up aging. After only about 10 or 12 months, the ICE mice had a frailty index that looked like a normal 24-month-old mouse.
They basically aged a year in a few months.
They did, just by stressing that organizational capacity.
Okay, so now for the hope. Reversing it. If the software is corrupted, the hypothesis is that our cells must have a backup copy somewhere.
That is the absolute core of rejuvenation research. We know that cells follow this path when they specialize. It's called Waddington's landscape. Think of a ball rolling down a hill into a valley. To rejuvenate a cell, you have to push that ball back up the hill.
And that's what epigenetic reprogramming does.
Exactly. Using factors like OSK, that's OCT4, SOX2, and KLF4, you can literally push the cell back up the landscape, restoring youthful gene patterns and measurably reducing its epigenetic age.
So let's dive into those technologies. The gold standard for this reset is genetic reprogramming with those Yamanaka factors, OSK.
Right, through transient cyclical expression, and the results in neuroscience have been just stunning. They successfully cured blindness in mice by restoring aged retinal neurons.
It's not just patching damage, it's regrowing youth.
It's regrowing youthful function, yes. And they did similar work with human brain organoids that had Alzheimer's mutations.
What happened there?
The OSK treatment restored organized electrical activity and reversed some of the key signs of dementia in these mini-brains. They also showed it could improve the pathology in a mouse model of tauopathy.
For listeners, tauopathy is basically the tangles of protein inside brain cells that are a hallmark of Alzheimer's.
That's right. And the treatment helped clear those up.
The key word here seems to be transient. You have to push the cell back up the hill, but not so far that it becomes a stem cell.
That would be bad. That could lead to a tumor. And that safety challenge is exactly what's driving the next big thing. Chemical reprogramming.
The safer version.
The safer, scalable, daily use version. The goal is to get the benefits of rejuvenation without using genetic engineering, and critically, without erasing the cell's identity.
How do they manage to do that?
They identified cocktails of small molecules that could do it. They used this really innovative high-throughput assay, the NCC assay, to distinguish between young, old, and senescent cells very quickly.
And the implication here is massive.
It's huge. We now know you can reverse aspects of aging with chemicals. With a pill, it moves the whole field away from complex gene therapies and towards something you could pick up at a pharmacy.
The good news is we don't have to wait for a full cellular reprogramming. there are things we can target right now.
Absolutely. We can target specific mechanisms of damage.
And one of the biggest targets is senescent cells.
Mm.
These zombie cells that stop dividing but just hang around secreting inflammatory stuff.
That inflammatory cocktail is called the SASP, and we have two main ways to deal with them: senolytics and xenomorphics.
Okay, break that down for us. What's the difference?
The distinction is really important. Senolytics, think of drugs like dasatinib or quercetin, are designed to selectively kill and clear out these senescent cells. They're the cleanup crew.
And xenomorphics.
Yeah.
They just try to quiet the cells down.
Exactly. Agents like metformin or resveratrol are xenomorphics. They don't kill the cells, they just modulate that inflammatory SASP. They basically tell the zombie cells to behave themselves without getting rid of them.
So the future is probably a combination of the two.
That's the consensus, yeah. The best results will likely come from cocktails: a senolytic to clear out the worst offenders, a xenomorphic to manage the rest, and maybe a metabolic enhancer thrown in.
Speaking of targeting, I was really struck by the discovery of this Gerazyme, 15-PGDH. It feels so precise, like finding one light switch that controls decline in a whole system.
It's a perfect example of precision aging medicine. A Gerazyme is just an enzyme that gets more active as we age. In this case, more 15-PGDH means less of a molecule called prostaglandin E2 or PGE2.
And PGE2 is critical for our muscles.
It's essential for muscle stem cell function. So when PGE2 levels drop, you get sarcopenia, age-related muscle wasting, and weakness.
So the intervention is beautifully simple. Just block the Gerazyme.
That's it. You inhibit 15-PGDH with a small molecule drug, PGE2 levels go back up, and that in turn boosts mitochondrial function, restores neuromuscular junctions, and you see this significant increase in muscle mass and strength in old mice.
It's hitting multiple problems with one solution.
One elegant solution, yeah.
Let's broaden out a bit to metabolic health. We hear a ton about sirtuins, these protective enzymes. But the sources remind us they're totally dependent on NAD+ bunker.
Right. Sirtuins can't do their job repairing DNA, stabilizing the epigenome without NAD+ load. The problem is our NAD+ levels just plummet as we get older.
So the idea is to use NAD+ boosters like NMN or NR to artificially raise those levels.
And support those critical anti-aging pathways, yeah.
This also ties back to metformin, which you mentioned as a xenomorphic.
Yeah.
The clinical work on this is really happening now.
It is. The TAME trial targeting aging with metformin is one of the most exciting studies going on right now. It's looking at whether this common anti-diabetic drug can actually promote healthy aging in humans.
So we have to connect all this amazing science back to what you, the listener, can do today, because our lifestyle choices are already modulating these exact same biomarkers.
Absolutely. And the research is getting very specific. With nutrition, for example, it's not just avoid sugar. It's about avoiding rapid blood glucose spikes. You might find a healthy food like grapes sends your glucose through the roof.
While things like dark leafy greens and Brussels sprouts have a measurably positive effect.
A huge positive effect.
And what about exercise?
The consensus seems to be shifting, especially for high-intensity interval training, HIRT. The new thinking is less is more. It's about getting that intense stimulus, but then allowing for adequate recovery so you get the benefits without the chronic stress and damage.
Which brings us to the biggest factor of all, maybe. Chronic stress.
It's an enormous aging factor. The analysis we looked at pointed out that people who identify as worriers or perfectionists put this really unique, damaging strain on their systems.
So what's the antidote?
It sounds simple, but it's gaining perspective. The researchers suggest that consciously comparing those daily worries to a genuine life trauma, like losing a loved one, can help reframe things. It can short-circuit that destructive stress response that erodes your epigenome.
This has been an incredible journey. I think the key takeaway for you listening is that aging science is moving fast, and it's moving towards precision medicine.
For sure.
We're not just waiting for things to break down anymore. We're getting these system-specific readouts of our biological age and developing these incredibly targeted ways to reset that corrupted cellular software.
And if we can really shift the medical system from treating diseases to treating aging as the root cause, the implications are just staggering. I mean, the research suggests that within a decade, it could be normal to get a blood test that tells you your biological age.
And a personalized prescription to fix it.
And a personalized prescription to bring those clocks back into line.
That is a future worth fighting for. So if the information theory of aging is right, if your cells really do hold a backup copy of their youth, we know you can tap into that today. So one specific action based on what you've heard, are you going to adopt to activate that internal reset button and boost your cellular resilience?
Thank you for joining us on the deep dive.
We'll see you next time.